as well as short questions. So the long questions will be talking about the components of the electron transport chain, the schematic representing, then description of the various complexes involved in the electron transport chain, then inhibitors and uncouplers. This is also an important topic for biological oxidation. Then your chemiosmotic theory, oxidative phosphorylation, what is it, how is it linked? So all of this will be covered in long question and answers. Okay, this is a very important topic and it is a very high yield topic and very frequently asked. Then in short answer questions also should get about like the various type of shuttles involved, the malate aspartate shuttle, the glycerol 3-phosphate shuttle. Then what is redox potential? You should know about it as well, because the entire concept of biological oxidation is based on redox potential. Then very important, that is the coupling of oxidation with phosphorylation, then the structure of ATP synthase. So all of this will be covered as short question and answers, and then of course the entire schematic representation and flow of electrons, that is the most frequently asked question in long essay types. Okay, so that was a brief introduction about biological oxidation and what all we'll be covering in this. Then talking about this, just like three different plans, the basic, which is for 3 months and comes at 15,000. Then the standard plan, which is for 6 months and comes at 30,000. And the mentorship plan, which is for 12 months and costs 50,000. So in the mentorship plan, you have personalized guidance as well as notes will also be provided. Then you can use my promo code as displayed on the screen to avail an additional 20% off. Then introducing you to the core features of Auricle. Here at Auricle, we provide you with live lectures so that you can clear your doubts in the moment. Then apart from that, we also provide you with recorded lectures so that you can assess any of the subjects as per your convenience, anytime, anywhere. Then notes are also provided based on the latest curriculum and CBME syllabus. Apart from that, MCQs, question banks are also provided by us so that you can assess the knowledge that you gained. Then talking about a very unique feature of Auricle, that is we have personalized counseling sessions here at Auricle. So apart from the academic support that we're providing, we're also willing to provide you non-academic support. So if there's anything you feel like you want to discuss or need some extra support and guidance, which does not necessarily be related to your academics, then also you can approach us. So feel free to book a slot with us anytime if you want some extra guidance and support from our end. We'll be glad to serve you that way. Okay, so now let's begin with today's lecture. That is biological oxidation. So you can consider that this biological oxidation is synonymous to a kind of a power plant, energy generating power plant in your body. So so that you're able to move, think, function properly. Okay, so this biological oxidation or electron transport chain is synonymous to a, you can for convenience purposes you can associate to a power plant that is generating a lot of energy. So all the functions of the body that are dependent entirely on this, so it is going to provide energy for the various functions that are going to take place in the body. So with that little introduction, let's begin. So the first question is difference between oxidative phosphorylation and substrate level phosphorylation. Okay, maybe you'll not be able to understand oxidative phosphorylation so well right now since this is the beginning of the lecture. But once you're done, I'll take this chart again if we have time. Then you will be very easily able to appreciate the differences here. Anyway, let's begin. So this is on this side, on the left side, we have substrate level phosphorylation. This one. And this is oxidative phosphorylation. So let's see what is the difference between the two. This is substrate level, this is oxidative phosphorylation. So substrate level phosphorylation, I'm very sure that you've already heard about this when we were taking different pathways like in glycolysis. Even in TCA, we came across substrate level phosphorylation, where I said that here at the level of the substrate itself, ATP is being formed. So, okay. So it says here substrate level phosphorylation refers to a type of phosphorylation in which phosphate group is transferred from substrate to ADP. Okay, so what is happening here in substrate level phosphorylation, that as and when the pathway is taking place, the products are changing into substrate. So from the substrate itself, a phosphate group is being attached, is being given or transferred to ADP to form ATP. So you see, ATP is being generated at that level of the substrate itself. You are not getting reducing equivalents which then need to go into the electron transport chain as an oxidative phosphorylation and which will then later on give energy. Okay. So that is what is substrate level phosphorylation. Now talking about oxidative phosphorylation, this refers to the type of phosphorylation which uses energy released from the electron transport chain to generate ATP. So this will be more clear once we're done with oxidative phosphorylation. That is what the topic is. So it is very much okay if you're not able to understand this right now at the moment, but at least understand what is substrate level phosphorylation. Okay. Then substrate level phosphorylation, I hope it's clear that the substrate is giving a phosphate to ADP and ATP is being formed. Whereas on the other hand, oxidative phosphorylation, what is happening? Reducing equivalents are being generated and then they go into electron transport chain. There's transfer of electrons, that is oxidation is taking place. And because of this oxidation, energy is being released and then that energy that is released, it is trapped or used to generate ATP. So that is oxidative phosphorylation. Now moving forward to the next point. So substrate level phosphorylation occurs in cytoplasm and mitochondrial matrix, whereas oxidative phosphorylation occurs in the inner mitochondrial membrane. Now, in this, a phosphate group is directly removed from a substrate by a coupled reaction and transferred to ADP. Okay. In oxidative phosphorylation, phosphate groups are added from the energy released in the electron transport chain. Then this is direct phosphorylation, this is indirect phosphorylation. Now this takes place in glycolysis. You know, there were two steps in glycolysis which were generating ATP in Krebs cycle as well. And this takes place in electron transport chain. Here NAD and FAD are reduced, whereas here what is happening to reduced equivalents, they are entering the electron transport chain, giving the electrons and themselves are getting oxidized. So it says NADH and FADH, they are being oxidized. Okay. Here 4 ATPs are produced, here 34 ATPs are produced. The change of the redox potential of the substrate is less. What is redox potential? It is the ability to gain electrons. Okay. The chain of the redox potential of the substrate is more. Then in substrate level phosphorylation, there is partial oxidation of the substrate. And here there is complete oxidation of the electron donors. So that was the differences between substrate level phosphorylation and oxidative phosphorylation. So now let's begin with oxidative phosphorylation so you will understand the differences even more clearly. Before that, we have a few examples to tell you about substrate level phosphorylation. So you can see here, this is 1,3-bis phosphoglycerate. This is a step in glycolysis. You can see it is changing into 3-phosphoglycerate. So as you can see here in the substrate, it is 1,3-bis phospho. There are two phosphates. Okay, two phosphates are there in the product. You can see there is only one phosphate at position 3. So there is one phosphate in the product that is being formed. So where is one phosphate going? So from this substrate, one phosphate is being transferred to ADP and ADP changes into ATP. So you see phosphorylation has taken place and this is substrate level phosphorylation because you're getting ATP at the level of the substrate and there is no involvement of electron transport chain here. Similarly, this other example, you can see phosphoenol pyruvate. So you can see there is a phosphate group in the substrate, but here in the product, you can see there's just pyruvate. So what happened from the substrate? One phosphate was transferred to the ADP to form ATP. Similarly, in acetyl phosphate, you can see what is formed here. Acetate is formed by giving this phosphate to ADP to form ATP. So these were a few examples of substrate level phosphorylation. Okay, so moving forward, let's talk about electron transport chain. So introducing you to that. Before that, let me give you a little concept of electron transport chain. That why is it taking place? When is it taking place? What is all the story behind this? Okay, so what is happening is whatever food you've had, okay, we're not plants. We cannot do photosynthesis and have our own food, synthesize our own food. So where do we get our energy then? Obviously, we need energy for the various functions taking place in the body. So we derive our energy from taking energy in the form of food. Okay, so we consume food, and then those food substrates, they are oxidized to give us energy, to provide us energy. So what is happening? The major macromolecules in your diet, they are proteins, carbohydrates, and fats. All of these ultimately converge into what? Acetyl-CoA. So carbohydrate, it is going to undergo glycolysis, form pyruvate. Pyruvate via link reaction is going to change into acetyl-CoA. Your fatty acids, they're going to break down into glycerol and fatty acids. Your fats are going to break down into glycerol and fatty acids, and your fatty acid will undergo beta oxidation and give you acetyl-CoA. Then your ketogenic amino acids, they will also give you acetyl-CoA. So ultimately, all the macromolecules in the food, they you will give you acetyl-CoA. So they're all converging at acetyl-CoA. Now this acetyl-CoA, it is going to enter TCA. Okay, it enters citric acid cycle. Now what happens? In this are generated. Okay, so you end up with NADH and FADH2. So reducing equivalents are generated, and then these reducing equivalents, they enter electron transport chain. They give their electrons to the various complexes in the electron transport chain. Oxidative phosphorylation takes place. The final acceptor of electrons is oxygen. Oxygen is going to get reduced to water, and you end up with water here. And the energy that is released because of the oxidation of electrons, that is transfer of electrons from one complex to the other, it is used to pump protons, and then a proton gradient is generated, and because of which there is ATP synthesis. Okay, so ATP is ultimately being generated. So this was an overview of the concept of electron transport chain. See, it says here the reducing equivalents from metabolic intermediates are transferred to coenzymes NAD and FAD to produce NADH and FADH2. Like I explained you that your acetyl reducing equivalents are going to be generated. Then these two reduced coenzymes, that is NADH and FADH, you're going to pass through the electron transport chain. And this passage of electrons through the electron transport chain is associated with loss of energy. So energy is lost, and because of this, the energy that is being generated, this is an exergonic process. So it is used to pump protons from the matrix into the intermembrane space. Okay, this will create a proton gradient. We'll study further, and then we will see how this is used to generate ATP. So a part of this energy is utilized to generate ATP from ADP. Okay, so that is what we've discussed about, an overview about electron transport chain. Now here this diagram shows us various complexes that are involved in electron transport chain. You have complex 1, complex 2, complex 3, complex 4, and ATP synthase, which is complex 5. So you have 5 different complexes from complex 1 to 5, and then you have 2 mobile carriers. As you can see here, this coenzyme Q and cytochrome C. So these are 2 mobile carriers, and then you have 5 fixed multi-protein complexes. So 5 fixed multi-protein complexes. That is 1, 2, 3, 4, and 5. These 5 complexes, and then apart from that, you have these 2 coenzyme Q and cytochrome C mobile carriers. So they are not fixed, okay, in the inner mitochondrial membrane. So the complexes 1 to 4 are carriers of electron, while complex 5 is responsible for ATP synthesis. This, as we proceed, you will see how that the first 4 complexes, they're involved in oxidation, that is transfer of electrons, and the last complex, that is complex 5, also known as ATP synthase. This is the complex which is involved in the synthesis or in the generation of ATP. Okay, now coming to the major chunk of biological oxidation. So this is very important. So we'll be talking about now the components of the electron transport chain one by one. We'll also discuss each of the complexes, then a schematic representation of the electron transport chain, that what the electrons and transfer, what is the electron flow, and in the end, we'll end by discussing the inhibitors and uncouplers, another very important topic associated with ETC. Okay. So beginning with electron transport chain. So what is electron transport chain? If you define it in simple words, it can be defined as redox couples arranged in ascending or increasing order of the redox potential. What is redox potential? It is the ability to gain electrons. So you have redox couple arranged in a series, as you can see, complex 1, 2, 3, and 4, in increasing order of the ability to gain electrons. Okay. So it says there is transport of electrons through the electron transport chain, and it is linked with release of free energy. And as your electrons are transferred from one complex to the other, there is release of free energy. The process of synthesizing ATP from ADP coupled with electron transport chain is known as oxidative phosphorylation. So you see, what is oxidative phosphorylation? What is oxidation? Oxidation is your flow of electrons, okay? Transfer of electrons to the various complexes, and that transfer of electron is linked to release of energy. Okay, so energy is liberated during this process. That is when electrons are jumping from one redox couple to the other redox couple. So what is happening? Your energy is being liberated. So the jumping of electrons is accompanied by release of free energy. And this release of free energy, it is then trapped to generate ATP. So this is known as oxidative phosphorylation. Then the complex 5 of the inner mitochondrial membrane is the site of oxidative phosphorylation. Why do we say that? Because your complex 5, that is the complex which is actually synthesizing your ATP. So I'll just write this here, that electron transport chain is a series of redox couple arranged in ascending order of redox potential. And what is redox potential? It is the ability to gain electrons. And redox couples, what are redox couples? They are those compounds which can exist both in oxidized as well as in reduced state. For example, your NAD plus and NADH. Okay, so it can exist as NAD plus, which is the oxidized state, and it can also exist as NADH, which is the reduced state. So this is going to form one redox couple. The other example can be your FAD and FADH2. Your FAD is the oxidized state and FADH2 is the reduced state. So here what I'm trying to say is that in electron transport chain, there is a series of redox couple that is arranged one by one in increasing order of the ability to gain electrons. Okay, so what will happen? There will be a spontaneous flow of electrons, okay, from couple 1 to couple 2 to couple 3. If I explain this further, I can give you another example. For example, this is one redox couple A and this is another redox couple B. And they are arranged in such a manner that the redox potential of A is less than the redox potential of B. Therefore, they are arranged in this manner that A is coming first and then B is coming. So what will happen? Electrons from A will jump to B because B has a greater or higher redox potential, which is it has a greater or more ability to gain electrons. Okay, so the ability of B to gain electrons is more compared to the ability of A to gain electrons. Therefore, electrons are going to jump from A to B. Okay, and this is basically the entire concept of electron transport chain. Okay, so it is based on this concept. Okay, now, so if you've understood this concept, now the electron transport chain. So I like focused, like gave a lot of time on making this concept very clear, because now the rest of it is very simple. So if you understand this concept, now electron transport chain is going to be very easy for you. We'll just quickly take the complexes now one by one, and everything will fall into place. You will catch up very quickly. So now talking about complex 1. So what is happening in complex 1? Your NADH dehydrogenase, that is one name for complex 1. The another name is NADH coenzyme Q oxidoreductase. That is the second name. What is happening? Your NADH is going to come and give its electron to complex 1. Your complex 1 is fixed in the inner mitochondrial membrane, as you can see here. This is the inner mitochondrial membrane. This is the complex 1. Your NADH is going to come. As you can see, it is going to give its electron to this complex 1. Then your complex 1, it has different components, FMN and iron sulfur clusters are there. So you see the electron from NADH is first taken up by FMN and then by iron sulfur complex, and then ultimately it is given to coenzyme Q. So coenzyme Q is going to take the electron from iron sulfur complex. So you don't have to like remember the various components inside the complexes. Okay, simply understand that your NADH is going to come, it is going to give the electrons to complex 1. That is more than enough. If at all you want to know, you can know that complex 1, the components are FMN and iron sulfur clusters. Okay, then from complex 1, coenzyme Q, which is a mobile carrier, it is going to come and take electrons. So what is happening? So NADH is getting oxidized and your coenzyme Q, it is getting reduced here. And therefore, it is also known as NADH Q oxidoreductase, because your NADH is going to get oxidized and Q, that is ubiquinol or coenzyme Q, it is going to get reduced. Therefore, this is another name. Then it contains iron sulfur complexes and SMN complex 1. These are the components of complex 1. Okay, and here NADH is going to get oxidized and your coenzyme Q is going to get reduced. So that was about complex 1. Moving forward to complex 2. Now your complex 2 is associated with an enzyme known as succinate dehydrogenase. So it has succinate dehydrogenase, and also one thing to note is that your complex 1 and complex 2, they're not arranged in a series. Okay, both complex 1 and complex 2, they are going to give their electrons to coenzyme Q. So what is happening? Succinate dehydrogenase, if you remember, this was an enzyme in TCA Krebs cycle. So succinate dehydrogenase, as you can see here in this diagram, it is going to form fumarate, and while it forms fumarate, it is going to give its electron to FAD, and FAD is going to change into FADH2. Now this FADH2 is going to give its electron to complex 2. This is complex 2. Okay, now from complex 2, the electrons will be taken by coenzyme Q. So that was about complex 2, that your succinate dehydrogenase, it is a part of complex 2. Then complex 2, it receives electrons from succinate and give it to coenzyme Q. So that is, in short, that succinate is going to give its electron to coenzyme Q, but I've explained it how it's going to happen. Succinate is going to form fumarate. This is going to happen by succinate dehydrogenase, and in this process, the electrons are taken by FAD, and then it forms FADH2. Then FADH2 is going to give its electron to complex 2, and complex 2, what are the components? It has FAD and iron sulfur clusters, as you can see, and then ultimately what is happening? Coenzyme Q is going to take these electrons as well. So you don't have to remember all this. You only have to remember that succinate, in short, is going to give its electron to coenzyme Q. So your succinate is being oxidized, and whereas your coenzyme Q is being reduced. Okay, therefore it is known as succinate Q oxidoreductase or succinate Q reductase, the name of this complex, that is complex 2. Okay, and its component includes FAD, and it also has iron sulfur clusters. Okay, now talking about complex 3. So your complex 3 is also called cytochrome B1 complex. So this is the third complex in the electron transport chain. Now what are the components of it? It consists of cytochrome B, cytochrome C1 and resky iron sulfur clusters, okay, or complex proteins. So these are the components of complex 3. Now what happens? Your electron from coenzyme Q, they are going to be transferred to complex 3 C. Okay, so that is the transport. If I draw it more simply, so you can understand, this is your coenzyme Q, and your coenzyme Q, it was getting electrons both from complex 1 and also from complex 2. Okay, now what happens? This coenzyme Q, it is going to give its electron to the electrons are given to complex 3. So here, this is complex 3. It is getting electrons from coenzyme Q. And this complex 3 is then going to give it to cytochrome C. These electrons are given to cytochrome C. Cytochrome C is another mobile carrier. So your cytochrome C is going to get reduced. Okay. So your coenzyme Q, it is getting oxidized here. Coenzyme Q is giving its electrons to complex 3 and complex 3 is giving it to cytochrome C. So your coenzyme Q gets oxidized and your cytochrome C, this gets reduced. Okay. Therefore, complex 3 is also known as Q cytochrome C oxidoreductase. Okay. And then we've also discussed about the components that it has cytochrome B, cytochrome C1 and resky iron sulfur complex. Then talking about complex 4. So this complex 4, this is the final protein complex in the electron transport chain and is named as cytochrome C oxidase. Okay, and is commonly labeled complex 4. So your complex 4 is also known as cytochrome C oxidase, and this complex 4, it is going to receive electrons from cytochrome C, and then this complex 4 is going to give it to oxygen. Okay, so the electron carrier cytochrome C transfers single electron, but multiple electrons are required for handling oxygen. So what it's trying to say is, in order to completely reduce oxygen, you need to have 2 electrons. Okay, if oxygen is receiving 1 electron, it is going to form superoxide. Okay, so therefore, in order to completely reduce your oxygen to water, you need 2 electrons. So what is happening? Cytochrome C, it is going to give its electron to complex 4 and then complex 4 is going to give it to oxygen. So your cytochrome C is getting oxidized. As you can see here, cytochrome C is giving its electron to complex 4 and then complex 4 is giving it to oxygen. So oxygen is getting completely reduced to water. Water is formed here, okay? So your cytochrome C is giving its electrons. So cytochrome C gets oxidized. And what is getting reduced? Oxygen is getting reduced to water. Okay, so your oxygen is reduced. Now complex 4, as we've already said, it is known as cytochrome C oxidase. Now, what is the final acceptor of electrons here? So the final acceptor, electron acceptor, is your oxygen. Okay, so oxygen is the final electron acceptor. Always remember that. And talking about the components of this complex, that is complex 4. So it has various components like copper A, copper B centers it has, then it has heme A3 centers. Okay, so these are the components of complex 4. Copper A, copper B, you can see here in this diagram as well. And heme A3 center, also known as cytochrome A3. Now, talking about complex 5 or ATP synthase. So, before I talk about this, let me, there's, okay, yeah. Let me now just give you recapitulate what we've already discussed. Okay, so we've discussed the oxidative phase of electron transport chain. So you see here, this NADH was coming. NADH was giving its electron to complex 1. Okay, so NADH gives its electron to complex 1, and then from complex 1, these electrons are taken by coenzyme Q. So as electrons jump from complex 1 to coenzyme Q, what is happening? This complex 1 is releasing energy, and that energy release is used to pump protons into the intermembrane space. So you can see 4 protons are pumped by complex 1. Now your second complex, let's talk about complex 2. This is receiving electrons from FADH2. So FADH2 is going to give its electron to complex 2, and then this complex 2 is going to give its electron to coenzyme Q. But here the energy release is not sufficient, that so is not enough to for complex 2 to act as a proton pump. So proton pump, complex 2 is not acting as a proton pump. Protons are not being pumped into the intermembrane space by complex 2. Okay, remember this. Complex 1 was pumping 4 protons into the intermembrane space from the matrix, whereas your complex 2 is not pumping any protons. Okay, now what happens? So your coenzyme Q, it has received electrons both from complex 1 and complex 2. Now coenzyme Q, it is going to give its electron to complex 3. As you can see here, complex 3 is again acting as a proton pump. Energy release is used to pump 4 protons into the intermembrane space. Now from complex 3, the electrons are taken up by cytochrome C, another mobile carrier. Cytochrome C is going to give its electron to complex 4. Complex 4 is acting as a proton pump as well, and 2 protons are being pumped this time. Now complex 4 gives the electron to oxygen. So oxygen is the final electron acceptor, and oxygen is completely reduced to water. So until now we've discussed this much of electron transport chain. Okay, now let's talk about complex 5. Okay, here. So moving forward, talking about complex 5 now. So your complex 5, it is also known as ATP synthase. So what is happening here? As protons are being pumped from the matrix into the intermembrane space, so a potential gradient is being generated. Okay, potential difference is created due to pumping of protons from the matrix into the intermembrane space, as your protons are accumulating in the intermembrane space, and this is known as proton motive force, and this proton motive force is used to drive ATP synthesis. Okay, so protons return to the, since the positive charge in the matrix is less as compared to the positive charge in the intermembrane space, since so protons are being pumped into the intermembrane space. So a potential difference or a gradient is being generated. Because of this potential gradient, the protons, they have a tendency to come back into the matrix. Okay, so the protons return to the mitochondrial matrix to an integral membrane protein, which is your ATP synthase or complex 5, okay, known as ATP synthase. Now, this ATP synthase is a multi-subunit complex that binds ADP and inorganic phosphate and converts them into ATP. This will take along how phosphorylation is taking place when we talk about the structure of ATP. But for now, what it is trying to say is that because of the pumping of the electrons from the matrix into the intermembrane space, a potential gradient has been generated. And since there is higher concentration of protons in the intermembrane space as compared to that in the matrix, so they have a tendency to move back, the protons have a tendency to move back into the matrix again. So when they return back into the matrix, your ATP synthase, it is going to act as a proton channel, okay, and this will in turn is going to synthesize ATP. How is that going to take place? We'll just discuss when we talk about the structure. So proton transfer is coupled with ATP synthesis, though your protons are being transferred back into the matrix, which is coupled with ATP synthesis. Okay, this is called as chemiosmotic theory of oxidative phosphorylation. ATP is not synthesized unless there is simultaneous transport of protons across the inner mitochondrial membrane. So your protons that were pumped via the various complexes into the intermembrane space, they are then traveling back into the matrix via the complex 5, and this pumping back or transporting back of protons across the across the mitochondrial membrane into the matrix, it is coupled with ATP synthesis, and this is known as chemiosmotic theory. Okay. So that was about the concept of oxidative phosphorylation. Now let's talk about the structure of ATP synthase or complex 5. Okay. So your ATP synthase or complex 5, it has two subunits. That is your F0 subcomplex and F1 subcomplex. Okay. As you can see here, this is the F0 in the diagram and this is the F1. So ATP synthase consists of two main parts or two complexes, F0 and F1. The F0, as you can see here, it is embedded in the inner mitochondrial membrane and is driving the hydrogen ions, or it is acting as a proton channel. So your F0, as you can see here or here in this diagram, here, you see F0 is present in the inner mitochondrial membrane, and it is going to, you can see it is acting as a proton channel, and your F1, it is present on the side of the matrix. Okay. And F1 acts as a catalytic site for ATP production. So F1 is involved in ATP production and your F0 is going to act as a proton channel. Now talking about the components of F0 and F1, your F0, it is made up of 10 C disc proteins, as you can see here. So there are 10 of these 10 C disc proteins. Okay. And these are hydrophobic and it is present in the inner mitochondrial membrane and it acts as a proton channel. Now talking about F1, as you can see here, so your F1, it has 3 alpha subunits, it has 3 beta subunits, and then it has 1 gamma, 1 delta, and 1 epsilon subunit. All of this is not shown in the diagram. Some of it is represented here because it is coming on the back side. You can see here gamma is shown here. So gamma basically forms the bent axle, and gamma, it is connecting F1 and F0. And then you can see beta shown here, then alpha shown here. So it has 3 alpha, 3 beta subunits, and it has 1 gamma, 1 delta, and 1 epsilon subunit. And remember that gamma is important because gamma is connecting F1 and F0. And also what is important about gamma is that it is acting as the rotatory subunit. Okay. This gamma, it is acting as the rotatory subunit. So the entire ATP synthase is not going to rotate. The gamma subunit is going to rotate, and it is also connecting your F1 and F0. So that was about the structure of ATP synthase. I'll repeat it again. Your ATP synthase, it is complex 5. It consists of two components, F0 and F1. Your F0, it is made up of 10 C disc proteins, and then it is present in the inner mitochondrial membrane. It is hydrophobic and it acts as a proton channel. Then talking about F1, it has 9 different subunits, 3 alpha subunits, 3 beta subunits, then 1 delta, gamma, and epsilon subunit. The gamma subunit, it is the rotatory subunit. That is the unit which is going to rotate. The beta subunit is involved in acting as the catalytic site. It is involved in ATP synthesis. I'll just talk about that. And also your gamma subunit is connecting F0 and F1. Okay, so now what happens is, how is this going to take place? That is your ATP synthesis. How is that going to take place? So what happens, because of the potential gradient that is generated, your protons, they are going to enter back into the matrix via the F0 subunit, and while they are being pumped back into the matrix, what happens, your F0 rotates along with the gamma subunit, and because of this rotation of the gamma subunit, there's conformational change in the beta subunit, and because of which the 3 beta subunits, they convert ADP into ATP. So you see how phosphorylation or ATP generation has taken place. The proton is pumped back into the matrix. I'll repeat myself again. Via the F0, your proton is pumped back into the matrix, and because of which your F naught, that is the 10C disc proteins, along with the gamma subunit of the F1, it is going to rotate. And as the gamma subunit of the F1 rotates, there is conformational change in the beta subunits, and because of that conformational change, the three beta subunits, they convert ADP into ATP, and your ATP has been synthesized. So that was about the structure and the synthesis of ATP. Now, components of the electron transport chain, just a summary. We've already discussed all the components and the complexes. The complex one, as I told you, it has FMN and iron sulfur clusters. It was receiving electrons from NADH. So yeah, NADH, your reduced NADH was giving it electron to complex 1 and itself was getting oxidized. Then from complex 1, electron was received by by coenzyme Q. Your succinate was changing into fumarate via the enzyme succinate dehydrogenase. So your succinate, it is going to give its electron to FAD. FAD was forming, then changing into FADH2. FADH2 was giving its electron to iron sulfur complex present in the complex 2, or in short, FADH2 gives its electron to complex 2, and then from complex 2, the electrons are taken by coenzyme Q. A coenzyme Q is going to give its electron to complex 3. And what are the components of complex 3? It is cytochrome C1, iron sulfur complex, and B1. Then what happens? To complex 3 gives its electron to cytochrome C. Then cytochrome C gives its electron to complex 4. Complex 4 has various components, that is copper A, copper B, cytochrome A3, also known as heme A3. And then finally, complex 4 is going to give its electron to oxygen, and oxygen is reduced to water. And along with this, I also told you that complex 1 was pumping 4 protons from the matrix into the intermembrane space. Complex 2 was not pumping any electron. It was not acting as a proton pump. The energy released by the transfer of complex 3 was also utilized in pumping protons, and it pumped 4 protons, and complex 4 was pumping 2 protons. Okay. And then because of this pumping of the protons from the matrix into the intermembrane space, what was generated? A potential difference was generated. Then that potential difference, it was known as the proton motive force, and it was utilized in generating ATP. How ATP was generated? It was generated via complex 5. The protons, they were entering back because of this potential gradient into the matrix via the F naught of complex 5. Because of this pumping back of the protons, what was happening? Your gamma subunit of the F1 of complex 5 was going to rotate. This brings a conformational change in the beta subunits of the F1, and because of which ATP or ADP was converted into ATP. So that was the summary and the components of the complexes once again. So we've discussed all that. So now let's talk about the sites of coupling with oxidation coupling with phosphorylation. So what are the sites where this is happening? There are 3 exogonic sites in the electron transport chain that result in the synthesis of 3 ATP molecules. So we said that ATP synthesis was taking place when there was simultaneous pumping of the protons. Okay, so pumping of the protons was taking place at complex 1, complex 3, and 4. So oxidation of FNH2 by coenzyme Q, this is complex 1. Then oxidation of cytochrome B by cytochrome C1, this is complex 2. And cytochrome oxidase reaction, this is complex 3. Okay, so these are the sites where your oxidative phosphorylation is taking place. So each of these reactions serve as coupling site for ATP production. Now, chemiosmotic theory we already discussed. Let's again have a look at what the notes have to say. The chemiosmotic theory explains the mechanism of oxidative phosphorylation. The electrons are transported along the components of the electron transport chain accompanying proton release, as we've already discussed. Once again, they've shown here in this diagram that as electrons, they are moving. You can see the path of the electrons through the various complexes, ultimately being received by, okay. So it says, when electrons are transported along the components of the electron transport chain, it is accompanied by proton release. So the part of the energy, free energy harvested during the electron transport chain, it is used to pump protons. The resulting uneven distribution of the protons generates a pH gradient, as we've already discussed, and a charge gradient across the inner mitochondrial membrane. This potential electrochemical potential energy generated by this gradient is known as proton motive force, and the return of the protons to the mitochondrial matrix is coupled to ATP synthesis. So a potential difference is being generated. The matrix is less positive as compared to the intermembrane space because it has more protons. So obviously from a higher concentration, they have a tendency to get back into the matrix, and that return of the protons to the mitochondrial matrix was coupled to ATP synthesis. And this was taking place with the help of complex 5 as seen here. See, there is a high proton concentration here on this end, that is the intermembrane space, and here this side is the matrix. So what is happening? Protons are being pumped back into the matrix, and this was coupled with generation of ATP. So this is your chemi-osmotic theory. Now what is redox potential? As we've already discussed, redox potential, it is the ability to gain electrons. Okay, in oxidative phosphorylation, the electron transfer potential of NADH and FADH2 is converted into phosphoryl transfer potential of ATP. Okay, so this is what is happening. जो NADH, FADH2, you reduced equivalents, they were coming and they were transferring their electrons. Okay, so their electron transfer potential is converted into phosphoryl transfer potential of ATP. So the standard reduction potential is a quantitative measure of the ease with which a compound can be reduced or how readily it accepts electron. What is oxidation? Oxidation is loss of electrons, and reduction is gain of electrons. So your reduction potential, it is a quantitative measure of the ease with which a compound can be reduced. Can be reduced means it can gain electrons. So I've already told you what is redox potential. In simple words, just remember it is the ability to gain electrons. So it is a quantitative measure of ease with which a compound can be reduced, or in simpler words, I've simplified for you. Just remember that redox potential is the ability to gain electron. So more the redox potential, more is the ability to gain electron. So more positive the standard reduction potential, more readily the compound accepts electron. So just remember that what is redox potential? It is the ability of a compound to gain electron. So more the redox potential, more the ability of a compound to gain electrons. Okay. Now moving on to inhibitors and uncouplers. So these are, like, you just have to remember the names. Complex 1, the inhibitor being rotenone and pyricidine. Complex 2, it is carboxyl. For complex 3, the inhibitor is antimycin A. For complex 4, the inhibitor is cyanide, azide, carbon monoxide, and ATP synthase. That is complex 5. The inhibitor is oligomycin. And the inhibitor for ATP ADP translocase, that is, this is the transporter for ATP ADP. It is atractyloside. Actually, the inner mitochondrial membrane, it is impermeable to ATP ADP. So ATP ADP, in order to cross the inner mitochondrial membrane, it needs a transporter, and this transporter is ATP ADP translocase. So your atractyloside, it is going to inhibit this transporter, and when this transporter is inhibited, what will happen? Your ADP will not be able to enter the matrix. Okay, and so what will not happen? Your ATP synthesis will eventually not happen. Okay, now talking about uncouplers. What are uncouplers? They make holes in the inner mitochondrial matrix, and this dissipates the potential gradient that is generated. So what is happening? Phosphorylation. Okay, so phosphorylation is not taking place. Uncouplers, they inhibit, they uncouple the electron transport chain from oxidative phosphorylation. So your electron transport chain, that is oxidation is intact, transfer of electrons is taking place. Electrons are transferred to oxygen to generate water. However, what is happening? Uncouplers carry protons across the mitochondrial membrane, making it leaky for the protons. So what is happening? The protons are they going to make alternative pathways, or they're going to make some of them, they are channel formers like your ionophores. They're going to make proton channels like your ionophore. It is a lipid soluble compound which makes the inner mitochondrial membrane permeable for potassium. So potassium is going to leak in into the matrix, and it is going to dissipate the gradient. And what will not happen? Your ATP synthesis not will take place. Or another uncoupler, as here it is given, 2,4-dinitrophenol. What is 2,4-dinitrophenol? This nitrophenol, this DNP, this is a weak acid. This is lipid soluble. It can exist both in a neutral state and an ionic state. So in ionic state, it is going to take one proton, then it is going to cross the inner mitochondrial membrane. Once inside the matrix, it is going to release the proton. So in short, what is it? What is it doing? What is it doing? That it is dissipating this potential gradient that has been generated. So therefore, it says uncouplers, they carry protons, just like DNP, this 2,4-DNP. It carry protons across the mitochondrial membrane, making it leaky. So basically, they are dissipating the gradient that has been generated, okay? And therefore, ATP is not synthesized. So in the presence of uncoupling agent, energy released by the electron transport chain is converted into heat. So otherwise, what was happening? The energy release that was used to generate ATP. But in the presence of uncoupling agents, the energy release, it is converted into heat. This mechanism is used by hibernating animals to stay warm in winters, since they don't need ATP for anabolic processes while they are resting. So, to hibernating animals, and also in infants which have brown adipose tissue, they have thermogenin. That is also based on the same concept, that the energy release, it is not used to generate ATP, rather it is used to generate heat. So this is a diagrammatic representation of the various inhibitors of different complexes. Of complex 1, you can see pericidin A, rotenone. Then complex 2, it is inhibited by malonate. Then for complex 3, you can see antimycin A. Then for complex 4, all the gases, this is H2As, carbon monoxide, cyanides, all the gases are the inhibitors. So this is just a diagrammatic representation. Now, another important thing is that your NADH is not able to cross the inner mitochondrial membrane. Okay, so the electrons that it is carrying will not be entering into the matrix. So what happens? There are shuttles for this NADH to cross the inner mitochondrial membrane. So the glycolytic pathway results in the reduction of NAD to NADH. The NADH is oxidized to NAD by the electron transport change in the mitochondria. Now, the mitochondrial matrix membrane is impermeable to NADH. Thus, a transport system would be required to allow the entry of NADH into the mitochondrial matrix. So, the inner mitochondrial membrane, it is impermeable to NADH. So, for NADH to cross this inner mitochondrial membrane, we have various shuttles, okay? Let's have a look at these shuttles. So, we'll be talking about glycerol 3-phosphate shuttle here now. Have a look at this diagram. What is happening here? Dihydroxy acetone phosphate, it is going to change into glycerol phosphate. And while it changes into glycerol phosphate, it is going to give its electron to NADH is going to give its electron, and it is going to get oxidized to NAD. So dihydroxyacetone phosphate, it is going to change into glycerol phosphate. It is going to give its electron to glycerol phosphate, and in this process, what happens? Your NADH is being oxidized into NAD. Now, this glycerol phosphate can cross the inner mitochondrial membrane. Okay, this is the mitochondria, and this is the inner mitochondrial membrane. So, glycerol phosphate is able to cross this membrane. Now, again, in the mitochondrial matrix, what is happening because of the enzyme mitochondrial glycerol phosphate dehydrogenase. This glycerol phosphate is going to convert back or change back into dihydroxyacetone phosphate. While it changes into dihydroxyacetone phosphate, glycerol is going to give its electron to FAD and form FADH2. And then this FADH2 will give its electron to complex 2, and then electron transport chain will take place, and ultimately ATP is generated. So, but what do you see? The electrons of NADH, they were carried via glycerol phosphate, and inside the matrix they were given FADH2. So the reduced FADH2, thus in the muscle and the brain, even though 2 NADH are produced by glycolysis, actually 2 FADH2 are available for entry into the electron transport chain. Now you know why? Because the electrons, they are carried via glycerol phosphate, and when it reaches the matrix, there the enzyme mitochondrial glycerol phosphate dehydrogenase acts, and when glycerol phosphate changes into dihydroxyacetone phosphate, then it is going to give its electron to FAD, and you get FADH2. That is the reason. And also, where is this shuttle, the glycerol 3 phosphate shuttle running? It is taking place functional in skeletal muscle and brain. Okay, let's quickly read what the text has to say. NADH on the cytoplasmic side is oxidized to NAD with phosphate, and the enzyme is cytoplasmic glycerol 3 phosphate dehydrogenase. Okay, then on the matrix side of the mitochondria, what is happening? The oxidation of glycerol 3 phosphate back to dihydroxyacetone phosphate is catalyzed by mitochondrial membrane bound enzyme, that is glycerol 3 phosphate dehydrogenase, and this oxidation is coupled to reduction of FAD to FADH2. Okay, so that was one shuttle. Now talking about the malate aspartate shuttle. This is another shuttle. Now you can see here, your aspartate, it is going to change into oxaloacetate. Now oxaloacetate is going to change into malate, and in this process, what is happening? Your NADH is getting oxidized to NAD. So the electrons from NADH2, they are being transferred into malate, and then this malate is going to enter into the matrix, mitochondrial matrix, via the inner mitochondrial membrane. So your electrons of the NADH, they have been carried by a malate. Inside the matrix, what is happening? The malate is going to again change back into oxaloacetate, and the electrons from the malate, they are taken up by NAD to form NADH. Then oxaloacetate is going to change into aspartate, and then aspartate is going to easily move out back into the cytosol. So that was about malate aspartate shuttle. And this malate aspartate shuttle, it is functional in heart and liver. Here what happens in the cytosol, the electrons are transferred from NADH to oxaloacetate, forming NAD and malate. The enzyme is malate dehydrogenase. Okay. Then in the matrix, what is happening? Malate is oxidized to oxaloacetate by the enzyme of the TCA, that is malate dehydrogenase. This is coupled to reduction of NAD to NADH. Okay. So in this process, your NAD is being converted into NADH. So you're getting the NADH inside the matrix now. So oxaloacetate is converted to aspartate by accepting an amino group from glutamate. The reaction catalyzed by so this is talking about how your aspartate is changing into oxaloacetate. So this is transamination reaction we've already spoken about. This is the keto acid for aspartate, aspartic acid. Okay. Then the aspartate readily crosses the mitochondrial inner membrane to enter the cytosol, and in the cytosol oxaloacetate. Okay, so with this we come to the end of the class. So that was all about biological oxidation or electron transport chain. We've covered everything that you need to know on this topic. If you have any queries, you can ask. I hope the concept of electron transport chain or biological oxidation is clear to you. And this is a very important topic, frequently asked, and like in essay type questions, and everything that we've discussed was important. All of it was important. Okay, from inhibitors to uncouplers, then the flow of electrons, then talking about the various complexes, how are the complexes arranged, the schematic representation of electron transport chain. All of this is important. So if you have any queries, send them before I end the class. Thank you for watching. Thank you.